Good evening, everyone. Thank you so much for joining us tonight for our latest medical sciences lecture titled Injury and Brain Cancer, Two Sides of the Same Coin. My name is Jo Kim Ho, and I'm a current medical student here at UCL and your chair for today's event. In today's fascinating lecture, Professor Simona Parrinello will talk about the connection between injury and brain cancer, glioblastoma, and the new avenues for intervention. We want to keep these lectures as interactive as possible, so please use the Q&A function on Zoom to submit your questions for our speakers. The hashtag to use on Twitter for this event is hashtag FMSLectures. This lecture will be recorded, and a link to the recording will be sent out via email to all the attendees after the event. Before I introduce our speaker, we would like to know about our audience. So we'll run a poll asking you to select the group you consider you belong to. Okay, thank you so much to all who voted. I can see over here that 43% are prospective students and their families, followed by the other group, which are undergraduate students. Okay. Yeah, thanks once again. And I'm delighted to now introduce our speakers. We have Professor Simona Parrinello. The professor is a professor of neuro-oncology at the UCL Cancer Institute, which she joined in 2018. She also leads the Samantha Dixon Brain Cancer Unit and co-leads the CRUK Brain Tumor Center of Excellence. Professor Parrinello's research focuses on adult neurogenesis and brain cancer. In particular, her group studies the cellular and molecular mechanisms that control neural stem cell behavior and how these mechanisms become deregulated to drive tumorigenesis in glioblastomas, with a focus on the role of the microenvironment. Thank you, Simona, for joining us today. Now over to you.
Thank you very much, Joachim, for the kind introduction, and thanks to everyone who's joined us for this session tonight. So I'd like to tell you a little bit about our research this evening, which, as you just heard, focuses on glioblastoma. So glioblastoma is a primary brain tumor that occurs in adults. And by that, I mean tumors that form from cells within the brain itself. And every year in the UK, approximately 12,300 people are diagnosed with a brain tumor, making brain tumors the 9th most common cancer in the UK. Of all these tumor subtypes, about a third are malignant tumors. And within this group, glioblastoma is the most common. And it's not just the most common, but it's also the most aggressive form of brain tumor. It's a very devastating disease. And even though survival rates vary from patient to patient, on average, the outlook for these patients, the prognosis is very, very poor. And even though standard of care treatments do extend survival in patients, they're not curative. And sadly, this prognosis has really not improved over the last 20 years. So it is clear that we have to do a lot better, and we need to develop improved treatments for this disease. And the way we approach this problem in my lab is that we believe that if we can increase our understanding of the biology that underpins glioblastoma, we should be able to harness this knowledge to develop more effective treatments. So we can't really begin our talk about glioblastoma unless we first talk about a very interesting type of cell, which is called the neural stem cell. And neural stem cells are very important players during brain development. They are the cells that form the brain during gestation. So neural stem cells are immature cells that are able to make more of themselves, but they also give rise to the three main types of cells that make up the brain. And these are specialized cells, mature cells, that are called neurons. I'm sure most of you are familiar with what neurons are, but just as a quick recap, they are electrically excitable cells that constitute the building blocks of the brain, and they function by communicating with one another and forming neural circuits. Astrocytes is the second type of important brain cell, and these cells have a variety of roles in the brain, among which is their support of neurons and neuronal circuits themselves. And finally, the third key cell type that is made by neural stem cells are the oligodendrocytes. And these are highly specialized cells whose function is to wrap around the very long projections that the neurons make, which are called axons. And through these projections is how neurons contact other neurons. And oligodendrocytes wrap around these extensions to form a layer of insulation, which is called the myelin sheath. And this is a fatty layer that works a little bit like the plastic that surrounds electrical wires. And the function of this system is to make electrical conduction along the nerves really fast. Can you see my pointer? Maybe I should do this. Okay. So for a very long time, it was believed that neural stem cells exist during development, and this is it. And that actually, when we're born with a certain number of neurons, and this number doesn't really change. And this was largely based on very fundamental work carried out by a very famous neuroscientist that was one of the fathers of neuroscience, Ramon Cajal, who spent a lot of his time looking at brain tissue and looking at neurons and describing them and drawing them in these very beautiful pieces of art, really, that he made. And from this work, he concluded that we're born with a certain number of neurons within the brain, and these can die over time, but they will not be replaced. And this has been known in the field as the Harsh decree. And this has been believed by scientists for a very long time. However, this dogma has begun to be challenged towards at the end of last century, starting from the late 1960s. And really, a series of studies by very important neuroscientists begin to understand that actually the Harsh decree maybe was not completely correct. And this was also based on very beautiful and interesting work on the songbird, which is shown here. And this was work that showed that the way songbirds learn new songs during the mating season is because new neurons are formed within the brain that go and integrate within existing circuits. And these are necessary for the birds to remember the new songs. And after that, additional discoveries were made to show that new neurons are formed also in the adult brain of mammals, including in humans. And this is because we now know that actually neural stem cells do not disappear after birth, but actually are still maintained not all over the brain, but in two specialized regions of the brain, between the two of which the subventricular zone is the most important and the most relevant for glioblastoma. And in this region, stem cells are still found throughout adult life. And here in a normal, healthy brain, what they do is they divide to make more of themselves, but they also give rise to neurons and to the two types of cells I described, oligodendrocytes and astrocytes, although a lot less. These cells together are called glial cells. But what was even more exciting was the discovery that while this process goes on at all times in the normal brain, this process also responds to injury. So if a brain is injured, for example, through a stroke, and these cartoons, by the way, represent a mouse brain on which the mouse has been used a lot as a model for these studies. After a stroke, neural stem cells activate even more, so produce even more of themselves and their progeny. And what we know now is that both neurons and oligodendrocytes formed in this region are able to begin to migrate towards the site of injury, where they then act to repair the brain to some extent, to replace the neurons that are lost as a result of the injury, and to repair area of the brains that have lost their myelin by forming new oligodendrocytes that will wrap around the axons and form new myelin sheaths. And so, of course, this has been a very exciting discovery in the field that has given rise to a lot of research, because we know that although these processes do exist, they're not very efficient, but there is great hope in the field to be able to improve these natural processes by understanding it better in order to help patients that have experienced an injury and regain function more efficiently. Another important discovery that has added to our knowledge of brain repair has been made even more recently. And this is that in addition to these two reservoirs of neural stem cells that we now know exist in the adult brain, there is a potential additional source of neural stem cells within the adult brain. And these are the astrocytes, the cells that I described to you at the beginning. We know that at least in some parts of the brain, particularly in a region called the striatum, when there is an injury, astrocytes can go back in their development and go back to a more mature neural stem-like state in which they can make more of themselves and begin to produce neurons. And again, this has been a very important finding in the field that has potentially revealed a new avenue for improving brain repair, because astrocytes through the brain could potentially represent a source of new neurons. So you might be wondering at this point, why am I telling you all of this and what this has to do with glioblastoma? Well, it has a lot to do with glioblastoma for two main reasons. The first one is that a lot of work in the field is beginning to reveal how neural stem cells very often can be the cells from which glioblastoma forms and originates. So if neural stem cells acquire mutations that are required for cancer to form, they can go on to give rise to glioblastoma. And this has been shown also in patients. In particular, very elegant studies have shown by comparing what mutations are present in brain tissue from patients looking at the tumor itself and the matched neural stem cells from these patients, they found that many of the mutations were shared, suggesting that in fact these cells could be the cells that give rise to the tumor. And furthermore, we know from a lot of work looking at the tumor tissue itself that in glioblastoma, once it's fully established, the tumors contain cells within them that are very similar to normal neural stem cells. They're not completely identical, but share many properties. So just like the neural stem cells, these cells that we call glioma stem-like cells can make more of themselves, but also are able to form more mature cells that are not fully specialized like the normal cells, but look a lot like neurons, oligodendrocytes, and astrocytes. And it is through this process of mirroring development, but in an aberrant manner, that glioblastoma develop and grow over time. And what this also suggests is that if the glioma stem cells are so similar to their normal counterparts, it is possible that they also retain the ability to engage some of these repair processes that I described to you just now that occur within the normal brain. So these are really the areas that we have become very interested in exploring within the lab. And in particular, I'd like to tell you a little bit about a project that we published last year, in which we focused on this very interesting behavior of the astrocytes, this observation that in some cases they can go back to an immature neurostem-like state. And this happens only in the context of injury, but if you remember, it doesn't happen everywhere. So we only in some types of astrocytes, for example, it does not happen in an area of the brain called the cortex. So we were interested in understanding whether this potential of the astrocyte to switch back to this neural stem-like state, from which potentially they could then go on to form a tumor, has something to do with the formation of a brain tumor in the first place. And we wanted to explore how this injury response might intersect with a mutation that might be present in an astrocyte. And in this case, we were looking at just simply one mutation, because we know that multiple mutations are required for cancer development, but presumably when the whole process kicks in, you would expect that only one or two mutations are accumulated to begin with. So we were asking whether the intersection between an astrocyte acquiring a mutation that may be relevant for cancer could intersect with this injury response, and whether this might drive the formation of a glioblastoma. And so to address this problem, we turn to mouse models, which we use in the lab a lot for our work. And to make a quite complicated story a bit more simple, what we did is we began by looking at mice in which we could label their astrocytes within the cortex with a red protein that is known as TD tomato. And you can see here in red, these are all astrocyte cells. And we asked what would happen if we took astrocytes that were completely normal, so had no mutations, and injured them, or if we took astrocytes with one single mutation that is relevant to brain tumor development, and then on top of that, we injured the astrocytes as well. And this is what we found. So you could see that in the context of the completely normal astrocytes, after an injury, not much happened. So the astrocytes are here. They have these beautiful ramified processes. And this is why astrocytes are called astrocytes, because they look a bit like stars. And after an injury, just because they're injured, they don't change their behavior very much. They remain like astrocytes. However, what we notice is that if we do the same thing in the presence of a single mutation, the astrocytes now begin to change and they become very rounded. And you can see this here in these images. So they no longer have their processes, they retract them, and also they begin to lose the expression, well, they lose some of the markers that are typical of astrocytes. So this suggests that the astrocytes are beginning to change, but they're not becoming neurostem cell-like. They're just a bit less astrocytic than they are in this context. And we could see that this response was very specific to the combination of a mutation and an injury, because we could detect these cells just in this group of animals, but not in the ones where there were no mutations. And even more, I think, convincing was the observation that when we looked at where these rounded cells that derived from astrocytes, where they were found, we found that they were almost only found very close to the site of injury, suggesting that there was really a crosstalk or a synergy between a mutation and an injured brain environment. So this suggested that it was the beginning of a change. And so we asked, okay, this was an early time point, only a few weeks after the injury was experienced. So we asked what would happen if we now let the mice age and we waited a bit longer. And we waited quite a bit longer. We waited about 10 months longer following an injury. So these were animals that were subjected to the same exact thing. They had either no mutation or a mutation with an injury early in life, and then we waited 10 months following the injury. And what we found to our surprise was that now these cells that had begun to change at 6 weeks, at 10 months, they had changed even further, and they had begun to show markers of this neural stem-like state, which is this protein here in green and blue. So the cells are rounded, but they're now also showing markers of this neural stem-like state. And in addition, they're able to divide, make more of themselves. And in some cases, we found that they were even able to go a step further and begin to form neurons. And you can see this here from the shape of this beautiful neuronal cell with the long processes and the axons. So this really told us that even just one mutation in the presence of an injury microenvironment is able to change these cells towards a more immature state. And this state is a similar state to the state from which glioblastoma initiates. And so in this state in which the cells can divide, they would potentially be in a position to then acquire more mutations that then would lead to the cells forming a tumor. So this was very interesting. So we wanted to understand this a bit more. So what is it about the injury brain environment that might be contributing to this? And we thought a bit more about what aging means. So injury alone in a young animal doesn't seem to do very much. We need to wait a long time. And we reasoned that actually what is known about aging is that the level of inflammation within tissues increases as a result of the aging process itself. And in the brain, we can look at this by looking at a very specific type of cell, which is called the microglia, which is shown here in green. And these very nice arborized cells. And the microglia are the first responders of the cells. When there is an infection or an injury, the microglia switches to what we call an activated state, in which they respond to these insults. And this can be detected using a specific marker called CD68. So when there is a lot of CD68, the microglia are responding. And if you compare a young brain in which no injury has occurred, we see that this level of activation is very low. But in the aged brain, even before we injure it or anything happens, these levels are already increased basally. And you can see this here between young and old brains. And we're expecting this from work that has been published before. But the surprise came when we began to look at how aging and injury might interact. And this is shown here. So when we looked at the injury, the site of injury, very close to where the injury occurred in the young, we see that there is even more CD68, which is shown here. And we would expect this because we have given an injury to the animals. But this was contained to the injury site. And when we moved a bit further away from it, the levels of activation of CD68 were low. However, upon aging, so when we leave the mice to age longer after an injury in early life, this is quite different. So we see that immediately near the injury site, there is the same level of activity that we saw in the young animal, which was very surprising because it suggests that even after 10 months, this injury is not fully resolved, but actually inflammation persists. Not only that, but it spreads even further away from the injury site. And now a little bit further away from the immediate vicinity of the wound, we still see higher levels of CD68 here. So what this suggested to us is really that the injury in early life and the normal process of inflammation that occurs over time with aging are synergizing, creating a level of inflammation within the brain that is much higher than normal and potentially has a role in driving these astrocytes to an early tumor stage. And for us to really confirm this hypothesis and prove it within the lab, we had to do what we call a functional experiment. And so this is what we did. And we reasoned that if inflammation is the process that drives this response in astrocytes, we should be able to mimic this in a young animal. And what we did is we repeat our experiments using either normal astrocytes or astrocytes that had the one mutation and injured them, as we did before. And then, in addition to that, gave a drug to the mice that increases inflammation. And so, in a way, this is a way of mimicking what's happening with age. And so when we do this, when there are no mutations, the astrocytes again remain normal, not much happens. But now if we do this in the presence of the one mutation, now we can fully mimic the response that we see happens naturally upon aging, where the cells go back to this neural stem-like state. And again, this is only happening when there is a mutation at baseline, but in a completely young animal. So this confirms this idea that if you experience an injury early in life, this might change the environment of your brain in a way that upon aging, the levels of inflammations are so high that if some cells are present within the brain that have the potential to begin to change and begin the formation of a cellular state that might lead to tumors, then that this would be allowed by the inflammation, the inflammatory microenvironment or environment of the brain. And so if this is the case, then we should see some evidence of this happening in patient data. And so to ask this question, we collaborated with a colleague here at the UCL Institute of Health Informatics, Albina Lai, who consulted electronic health records from patients and asked the question whether patients that had experienced an injury to the brain in early life would be more likely to develop a brain tumor later in life than patients that didn't. And these numbers just represent the likelihood, the risk of that happening. And what she found is that indeed there was a greater risk in patients that had an injury in early life, of about 3.5-fold greater chance of them developing a tumor than patients that did not experience an injury, linking these two processes to each other. So I think if we put this together, what our work is in suggesting really is that in a healthy brain where inflammation is low, normal cells may acquire mutations, but in itself, this is not sufficient for them to change in any way or begin to form a tumor, because these cells are kept in their normal state by the normal brain environment. However, if we experience an injury and then on top of that, the aging process increases inflammation within our brain, this acts a bit like a perfect storm, and together with the mutations allows the cells to change and become more immature, more stem-like, and enter a stage in which if they experience more mutations, they acquire more mutations, they will be able to form a tumor. And the balance between healthy tissue and inflamed tissue plays a very, very important role. So far I told you about injuries to the brain that come from the outside, so external injuries, but there is a whole other aspect of injury that is very relevant to glioblastoma, and this is the injury that the tumor causes to the surrounding brain. And I think if we just think about how the tumors form, this is quite obvious. So a tumor that forms within the brain forms within a very confined space, which is encapsulated by the skull. So as the tumor grows, it will invariably compress the surrounding tissue, and this is likely to cause the surrounding tissue to become injured and damaged, for neurons to die, oligodendrocytes to die, for inflammation to take place. So in the lab, we're very interested in exploring this and in understanding what the crosstalk between the growing tumor and the surrounding brain that becomes injured does, what it means, and how it affects the behavior of the tumor itself and its growth. And so we began to look at that in a very specific context, which is the process of tumor spread. And to highlight this process and how important it is for the tumor, and actually it's one of the main causes why we cannot treat this tumor particularly well, I show you this image. This is one of our mouse models. And here again, the tumor cells are labeled with the same red protein that I showed you before, TD tomato. And you can appreciate that there is a main mass of tumor here, which we also call the tumor bulk. This is the main mass. However, the cells, and probably is the mass that would be taken out with surgery during treatment of the disease. However, you can see that many of these tumor cells have spread very extensively throughout the normal brain, which is here in blue. And this is what makes a resection, complete resection, impossible within the clinic. But as the cells spread within the brain, they like to spread along specific regions. And in particular, you might have noticed this very clear ribbon here that forms this C shape. And this is the white matter of the brain, which the cells like to use a lot to spread around. What is the white matter? The white matter is a part of the brain that is very rich in myelin, which is the substance that I described at the beginning of the talk. And these are areas where the axons that are surrounded by myelin come together in bunches and bundles. And the white matter really crisscrosses the entire brain, as you can see from this video that comes from the brain atlas, where all these tracts have been mapped in human. It's really beautiful, and it can really show you how extensive this network is. And because the tumor likes to go in these regions, these represent like natural highways for the tumor cells to spread and invade throughout the brain. But what we wanted to address in the lab was, what does this mean for the tumor cells? So when the tumor cells enter these regions and spread through them, what is the impact of this process on the tumor itself? And so we began to address this problem a few years ago. And again, we turned to a mouse model in which we generated a tumor. Here, in this case, the tumor cells are labeled in green with a protein called green fluorescent protein. So we let the tumor develop. And then what we did is we took the tumor cells out again, and we compared the molecular makeup of the cells that were found in the main mass of the tumor, the bulk, to cells that had been invaded either along the white matter or in regions of gray matter. And then we compared the molecular profile of the three regions. And this is shown in this plot here. So this is a PCA plot, and the details are not very important, but all you need to know is that if cells are similar to one another, they will group together in this plot. If they're different, they will group far apart. And you can appreciate, I think, that when we did this, the cells that came from the bulk of the tumor separated away from cells that were invading within the brain, regardless of where they were invading, suggesting that they were different. But what we found that was even more intriguing was that when the cells were invading within the white matter of the brain, they looked different from cells that invaded within the gray matter. And it turns out that the reason why they are different is because when the tumor cells go within white matter, they begin to look more like normal oligodendrocytes, the cells that I told you about, which are the ones that form myelin. So this makes sense because, of course, oligodendrocytes are very abundant in the white matter. There is a lot of them. And so this suggests that the tumor cells, when they find themselves in this white matter environment, they begin to behave more like normal cells. And consistent with this idea, what we found is that when the cells underwent this switch to becoming more oligodendrocyte-like, they were dividing cells less, and they were also spreading less, which is somewhat counterintuitive and suggests that these tumor cells were then less aggressive and less malignant in white matter. So this was very interesting because it also suggests that this could potentially be a weakness of the tumor that we could exploit for treatment. So we began to look at this in more detail. And it turns out that the reason why this happens is because the tumor cells find themselves in an injured white matter environment. So this is comparing the white matter in a normal brain versus the white matter in which tumor cells have invaded, infiltrated, and spread. And I think you can already see by eye that these are very different. So these are images taken at the electron microscope that allow us to see things in very minute detail and blow them up to a very great degree. And here, these gray circles are the axons. This is a cross-section through the axons, so we're seeing them as as if you were to cut a tree and looking down its trunk. And you can see here the axons are very regular, and they have these really dark black rings around them, which is the myelins. And this is beautifully organized. But when the tumors invade within these spaces, now the myelin becomes very disrupted. It's much thinner, the axons look darker, and they're much more irregular. And these are all signs of the fact that the tumor, the myelin here becomes injured and disrupted. And this is quantified here. You can see that in the areas that have tumor cells, we lose myelin and the axons are dying. So the tumor cells are finding themselves in an injured environment. And what was very exciting about this work is we also found that when this happens, when the tumor cells infiltrate within these regions, the normal oligodendrocytes that normally sit there and myelinate begin to die. So as more tumor cells invade, we see less and less oligodendrocytes, as shown in this graph. So they die off. And what happens is that the tumor cells seem to realize that, and somehow the injured microenvironment, the injured environment of the white matter is driving them to remember that they are a cell that is similar to a neural stem cell and is now trying to repair this injured environment. And to do that, it becomes more like an oligodendrocyte and begins to associate very closely with the axon. So the axons are here shown in red. They're these filaments, and you can see the green tumor cells are aligning next to them, and in some cases, they're even wrapping around the axons like a normal oligodendrocytes would do. And so in this way, the tumor cells that are trying to repair are, by default, becoming a little bit less malignant and a little bit more normal. So this is exciting because it means that the tumor cells have the potential to remember where they came from, and they have the potential to become less malignant and less aggressive. So the question we wanted to ask was, can we harness this normal response that the tumor cells have remember and still have within them for treatment? And so we reasoned that one way we could do this is go back to look at compounds and drugs that have been identified to be able to induce a normal neural stem cells to form an oligodendrocyte, something that would work in normal cells. And we asked, would something like that work in the context of a tumor as well? And in particular, we chose a drug that is called pralucast. And this was very exciting because this drug is already used in the clinic as an asthma medication. So we asked if we took a tumor in a mouse and we treated these tumors with either a control drug or with pralucast, would we see any difference? And the answer is yes, we did see a difference, because when we looked at how many tumor cells that were treated with pralucast were becoming more oligodendrocyte-like, we saw that there was an increase, suggesting that this drug, even in the context of a tumor, can promote these cells to take on a more normal behavior, a more normal oligodendrocyte-like state. And consistent with this, at the same time, because they were becoming more normal, the cells were also dividing less and the tumors were a lot smaller, suggesting that in fact there may be ways by which we can harness this normal process that the tumor cells still have and can activate with a treatment or a drug in a way that would be potentially independent of where the cells are in the tumor, and could be used as a general treatment for cells throughout the brain. So really what this story is telling us is, again, the same thing, that glioblastoma mimics brain repair. And what our data suggests is that when the tumors spread within the white matter, what they do is that they cause an injury to the white matter. As you can see here, the red, which is in this case the myelin, begins to disappear. And this injury to the myelin then talks back to the tumor cells, basically inducing behaviors that normal neural stem cells have and the tumor still retains, so that the tumor cells tries to repair this injured environment, and by doing so becomes less aggressive and less malignant. So just to put this all together, what I try to tell you today is that the brain has specialized cell types within it and processes that are there to help the repair of the brain. They're not very efficient, but they're still beneficial to contain and repair an injury. However, these processes also make the brain more vulnerable to tumor development. And for example, in the first story that I told you about, we found that normal processes that occur in injury can be hijacked during the early stages of tumor development. For example, when an astrocyte that experiences a mutation and an injury can go back to a neural stem-like state. In other cases, these processes could be beneficial potentially for treatment and slow down tumor growth, because in an established tumor, when the cells are exposed to an injury environment, they can remember their neural stem cell roots and use repair processes that ultimately make them less aggressive. But I think overall, what our work is showing is that the more we understand about the complicated links between injury and brain cancer, the more we will be able to identify new strategies that might lead to improved treatments. And I hope more generally, I convinced you that injury and brain cancers may really be two sides of the same coin, and that injury processes are intimately linked with brain tumor development and progression. And just before I take questions, I would like to make a couple of acknowledgements. The first one is just a nod to a person who has been very important in my life and was my PhD supervisor, Judy Campisi, and it's with her that I began the session about injury and cancer. And I just would like to acknowledge her because she recently passed away, but actually none of this work would have happened without her influence. And then obviously second, and importantly, the people in my lab, who are a fantastic team of scientists that are very dedicated and enthusiastic, and particularly two people who have really led the work that I discussed today. One is Holly Simpson-Rugdale, who has worked on the Astrocyte project, and the other one is Lucy Brooks, who has worked on the oligodendrocyte story, and alongside them, Mel and Wenhao have had really important roles in the development of these two projects. And with that, I'd also like to thank my funders and obviously all of you for your attention, and I hope you have many, many questions to ask. Thank you very much.
Thank you for such a fascinating lecture. Now to the Q&A session. So thank you to all of you who have submitted questions in advance of this lecture. I'll just begin with a selection. So my first question, or rather the audience's first question is, can it be assumed that early intervention after brain injury may help to delay the development of glioblastomas?
Well, I think we have to remember that a lot of what I showed you today is coming from experimental mouse models. So of course, we need to take this with a grain of salt, and there is still a lot to do before we can transpose these concepts to real life and to patients. But I think if we wanted to extrapolate from this work, the answer to the question would be yes. Our research suggests that if people that have experienced a trauma to the brain may constitute a risk group for the development of brain tumors, and therefore should probably be monitored more closely. And the more we understand about this process, the more we might find strategies to interfere with it, which could be preventative. And but of course, this doesn't mean that every single person who has experienced a brain trauma will develop a tumor later in life, because as I showed you, it's very complicated. It's not just about the injury. You also need the cells to have mutations, and one mutation alone is not sufficient either. You need to accumulate several mutations. But yes, I think to give a shorter answer, I think yes, it would be potentially beneficial to at the very least keep a closer eye on those patients.
Thank you. And another one. So currently, 1 in 2 people will develop some form of cancer within their lifetimes. Do you think there's a reason to believe that this statistic will become less worrisome?
That is it's a very difficult question. Of course, there is a lot of research that is happening across the different cancer types, and there is a lot of hope, and there's been a lot of success in many cancer types where survival rates have improved considerably. And I think many reasons to be optimistic. For example, all the advances that have been made in the field of immuno-oncology, where we're using the power of our own immune system to treat cancer. And I think we will continue to improve. And another area that I think we need to think about a lot more that will have a very big impact is prevention. What can we do to really stop tumors from forming in the first place? And I think a lot of research is sort of shifting towards that area because it's so important. It's the area where we can probably have the most impact as well.
Okay, and now I'll just move on to questions from this session. Do these ideas of brain injury, so the action and response of astrocytes, do they overlap or have any possible correlation with the activity and impacts of seizures?
We don't know is the simple answer. So the model that we used was a traumatic brain injury to the brain. And we know that astrocytes can switch back to a neural stem-like state after certain type of injury, such as stroke, for example, and excitotoxic substances, for example, the liver of toxic stimuli. So it is possible. I mean, we haven't really looked at that in detail, and I'm not aware of any specific research, but potentially there could be a link because, again, the inflammation that accompanies any type of immunotherapy, which is plays a very, very important role.
And one of the other questions, I think it's following from what you mentioned during the lecture. Couldn't the presence of CD68 in the distal of the age population already be present before the injury?
Yes, so we know that at steady state, before we even injure the brain, the age brain is more inflamed than the normal brain. So this has been known for a while, and it's not unique to the brain. Most tissues become more inflamed as we age. But what was interesting was that if we combine that basal process that happens anyway as a result of age with an injury, the level and extent of inflammation was even greater. So there is an additive effect between the basal level of inflammation that occurs as we age and the fact that the brain had experienced a trauma earlier in life. And what was very surprising was that this additive effect was maintained for such a long time, because we would have expected that following an injury, once repair has taken place, the level of inflammation goes down to nothing, or to a normal and injured brain, but that was not the case. The inflammation was maintained and persisted, and as if like there was a memory of this injury from earlier in life.
Another question, are Alzheimer's and other forms of dementia possibly linked to brain cancer or glioblastoma formation?
I don't really have the answer to that. I don't think there is much evidence in the data that would be the case. And it's not something we have looked at in detail, I have to say.
Okay. And another one, have you got any advice for medical students looking to start research in neuro-oncology?
Do it. Well, I'm not medically trained. I'm a biologist, so I'm not super familiar with how the career progression is in medicine, I would say. But I think there are definitely opportunities out there, particularly at later stages of the training. And there is really a great need for more neuro-oncologists, as well as clinical researchers who want to do a combination of sort of clinical practice and research in this area. So there are definitely opportunities out there, and there aren't enough people doing it. So if this is something that you're interested in, by all means, look into it and you know, come talk to me or some of my colleagues, because we need you.
Great. And are there specific mutations that make brain injury more susceptible to the onset of GBM?
Yeah, so it's a great question. So we only looked at one, which is a mutation in a gene that is very commonly mutated in all types of cancers, not just in brain cancer, and that is a gene called P53. And there we found a clear link, and I didn't really have time to go into a lot of detail, but we worked out also all the molecular mechanisms by which this happens, and we know why P53 is so important because of the way it communicates with injury signals within the brain, and it allows for the astrocyte to really go back to this neural stem-like state. We haven't looked at other mutations, so I don't know is the simple answer, but we found that a very important role for a specific signal, which is a molecule called EGFR, which incidentally is a gene that is normally amplified in brain tumors, meaning that one of the common mutations that happens very early in a GBM is that this gene becomes repeated multiple times within the genome. And so there are much higher levels of EGFR than you would have in a normal cell. So we know that this molecule is very important for early tumor development. So I would expect something that leads to the same effect, meaning an increase in activity of this EGFR molecule would probably have a similar effect, but we have yet to test that in real life.
Okay. And has there been any studies on the incidence of brain tumors in people playing sports where brain injuries can occur? So like contact sports, maybe. Yeah.
Yeah, that's a very interesting question. So this is something that obviously is very hot and you know, something that journalists go after a lot. And when we started this work, there were a lot of case studies published, some supporting a link between a prior trauma and brain tumor development, and some saying there were no links. And I think the problem is that the number of patients out there is quite low, because glioblastoma, even though it's the most common type of brain cancer, is still a relatively rare disease. So you simply don't have the numbers of patients to do this in a very robust way with the numbers that you need for the statistics. And so if you look at a small group of patients, you may not see any significance, but that doesn't mean there isn't. But there are definitely some case reports and some anecdotal studies that have that claim there's a link between contact sports and a higher incidence of brain tumors. But again, I'd like to stress that that doesn't mean that if you play high contact sports, you will get a brain tumor. It is potentially increases your risk, but it still depends on you having mutations in the right cells in the right time, and many mutations before a tumor actually does form.
Okay. And there's a question on, so the body regulates inflammation, but why doesn't the inflammation subside in young adults after brain injury?
Yes, I would love to know that myself. So it subsides to a great extent. So I only showed you the microglia response, but inflammation is more complex in that there are many other types of cells. So we know that many other inflammatory cells clear after injury, and also the response of the astrocytes themselves, because astrocytes are very important in containing the injury, and they form a scar around the injury. And that doesn't completely resolve, but it kind of goes down a lot over time. So all of those processes subside, but we were very surprised to see that the microglia response doesn't completely go back to normal even after a very long period of time. And we don't know that, but obviously that means that the consequences of a trauma or an inflammatory episode within the brain can be very, very long lasting, at least in rodents.
Okay. And another quite interesting question. Are there certain parts of the brain, if injured, are more likely to form tumors there? For example, areas closer to like the gray matter, like the cerebrum.
Yeah. So again, it's a great question. We only looked at this particular region, which is the cortex. And the reason why we did that is because normally these cells do not respond to injury. So they're quite resistant to this process of reversal to a neural stem-like state. So we wanted to know if having a mutation changes that, and we found that it did. So we haven't looked at other areas, but my guess would be because there are some types of astrocytes in other parts of the brain that do this reversal to neural stem cells, even if they're completely normal. I would expect that those cells, for example, in the striatum, which is an area of gray matter, would be even more prone to change and begin to get to a state that would be permissive to them be forming a tumor if the right mutations are acquired.
Okay. And so earlier on you mentioned the importance of a brain injury. Would cumulative shocks to the brain have a similar impact to the development of tumors compared to one larger in amplitude shock onto the brain?
What do you mean by shock? Like a trauma?
Yeah.
So yes, again, another very interesting question. We haven't tested that because it's quite a harsh thing, for an experimental protocol to do. But I would expect that probably yes. And the reason for that is because we're beginning to understand a bit more about how the tumor injuring the surrounding brain as it forms has an impact on tumor development. And there, actually, the fact that the tumor is constantly injuring the brain is very, very important. And so I would think that a chronic injury may be much more effective at driving tumor formation than an acute one, because an acute one is an isolated episode and it resolves at least to some extent. And even in our experimental models, we saw that there was this reversal to a neural stem-like state late in life, but we didn't see the cells forming tumors per se. So they only reached that stage in which they could potentially form tumors, but they didn't form a tumor. But I wouldn't be surprised that if we did the same thing, but made a chronic injury, now within the lifespan of the mouse, we would actually see tumors forming, because you have a constant and very heightened level of inflammation that is constantly there and provides all the signals and the environment that supports these cells and makes them divide.
Okay. Thank you for that. Is it possible that more active and healthy people who get a brain injury are less likely to have tumor cell spread?
I don't think I can really answer that, because it's also very sort of relevant to people. And as I said, our research was really done in mice, so one can only speculate, really. Can you repeat the second part of the question about the spread?
So I think that they were basically asking that if people kept more active and healthy, is it more likely to sort of reduce tumor spread?
I mean, it's very, very hard to speculate, I think. So I'm not sure I can comment on that one, I'm afraid.
That's fine. Um, and there's a question on are there currently any treatments towards preventing this tumor from forming in the future? And are there any methods of detecting the tumor at early stages before it gets serious?
Yeah, so that is, I think, a very big problem in this field. So I really believe that one way we can make a difference is that if we identify the ways and the mechanisms that are, lead to the formation of these tumors early on, we can interfere with those, and that is much more likely to succeed than trying to deal with a very late tumor that has evolved a lot, that is very complicated, that has, interacted with the brain environment in a way that supports it growth and so on, and that it's very that it changes all the time, so it becomes a very difficult problem and something that cannot be treated very well, or at least in a way that would target all the cells. So I think if we went a lot earlier, we would have a much better chance of stopping the cells in their tracks before they reach that late stage. However, in the context of GBM, this is a very difficult problem because we don't really have ways of detecting it very early. So imaging with MRI is the way to do it. But we know from the experience of our clinical colleagues that we begin to see a signal on the MRI quite late within the tumor evolution. So that means that by the time we detect it, the tumor is probably very late stage. However, I think there are some very exciting new developments in areas such as looking at identifying tumor cells, for example, in the cerebrospinal fluid or in the blood, that will, using markers that will give us a way that is relatively less invasive to begin to monitor whether or not there is evidence for these early tumors. And there, if these approaches become successful, then we now have a way in where we can begin to tackle this early disease stage. And so we need to really work on two fronts: work out the biology of the early disease stage and what are the weaknesses of these cells that we can target, and on the other hand, develop better ways to detect these early lesions. But I think we're making progress on both sides, so I am hopeful that we will get there in the end.
Okay, thank you very much for that. There are still quite a number of questions, but I'm afraid we've already run out of time, and we'll have to leave it there. However, if the audience could please provide us with some feedback about today's session, please do so by filling out the survey, which will be sent to you following the event, and that would be greatly appreciated. Right, and thank you again for all of your comments and questions, and thank you especially to Professor Simona Parrinello for such an amazing lecture. Have a great evening, everyone.
Thanks for having me. Thank you.